So I want to take a little different tact and talk to you about, as Catherine mentioned, small things, and particularly I'm going to talk obviously from my background of microbes, but I'm going to start with a very different premise, and this actually is a theological premise. So I don't usually talk theological to begin with, but if you know Timothy or 1 Peter or Isaiah, there's a statement that says all flesh is grass. All flesh is grass. And the question, and if you go into the theology of those statements in the Bible, what you find out is they were written to dealing with the short nature of human life, the importance of life, the fact that we're all going to die, and at some point in time, we all become grass and part of that grass. So having stated that premise, you have to look at what happens to all the carbon that we produce on this planet. So there's some recent calculations that on a yearly basis, there's about 224 petagigs of carbon that is produced on this planet on a yearly basis. And if you look at all the animals on the planet, this is all the insects, all the fish, all the birds, all the mammals, all the humans, whether we eat it primarily or secondarily, you come up with a calculation of somewhere between animals consume between 4 and 19% of all the carbon that's produced annually. Okay. Well, what happens to the other 81 to 96% of that carbon? Now, you all know that some of it sits around for a while. Okay? Some of it sits around for a while, some of it stays in place, but eventually all of that carbon, all of that flesh becomes grass. And the question is, how does that happen? And how it happens is why the fungi and bacteria are important. These are the organisms that contribute to the recycling of carbon on the planet. This is why, from my perspective, unless you look at how the microbes respond to climate, you don't really understand how climate's going to affect all the ecological systems on this planet, whether you're talking about oceans, freshwater, or terrestrial systems. And so from a microbial perspective, the carbon use falls into three compartments. You either are saprophytic, which is that middle picture of the puffballs growing on that log. You are pathogenic, which is the ringworm on the hand, or the organism, the ascomycete that's parasitizing the pupa underground. Or you are symbiotic, having mycorrhizal relationships with the roots of plants. Now, interestingly enough, we all know that these symbiotic relationships, for example, are very useful, and most trees, in fact, almost all trees need them. Almost all plants need a symbiotic relationship to acquire nutrients. What you may not realize is that the cost can be high. 40% of all carbon fixed by a tree goes below ground to the fungus. 40%. Now, and so back in about 1980, the foresters said, hey, why don't we eliminate the fungus and we can get that 40% back to the tree? Well, when they reduced the fungus, they didn't get the 40% back. They actually took what they had and decreased the total production by an additional 40% because the fungus is that necessary for the tree. If you like oranges, can't grow them without the symbiotic fungi. If you like apples, can't grow them without the symbiotic fungi. If you're cracking pecans or harvesting your pecans right now, they need the symbiotic fungi in order to help those trees grow. That's the nature of microbes and environment. You also know that recently we've discovered that our ability to stay healthy is dependent upon the gut microflora. And if you change the gut microflora, you become unhealthy. In fact, it's also been shown recently, this is interesting, that why do you go out and like to garden? Do you like the smell of fresh earth? And the question is, why? Well, it turns out there's a bacterium that produces that odor. And if you spray that bacterium on mice, you get higher levels of the hormones that elicit a good feeling in their brain. And you can watch that develop. So it's the microbes controlling us that allow us to do the things we do. So having said that, all of our work in our lab is focused on what role do microbes really play in natural systems, how do we account for their activity, and how do we understand how climate and human activity impact? So I'm going to give you sort of a brief synopsis of some of the things that we've been involved with, linking it to climate change, climate variability, and human activity. Well, interestingly enough, I started working on fungi as part of a program up in University of Calgary working on the oil sands. I worked on the oil sands looking at how to revegetate that whole system after you come through and do extraction of oil. But since I've been in Lubbock, a lot of our work has been focused at Big Bend National Park. And we were part of a series of what are called small referenced watersheds to understand how climate variability and anthropogenic inputs, particularly nitrogen, is driving ecosystems. So we had 11-kilometer elevational gradient in which we started. Big Bend was the driest reference watershed, and Olympic National Park was the wettest. So we had Olympic National Park, Noatak in Alaska, Great Smoky Mountains, Rocky Mountain National Park, Isle Royale. We were all collecting the same kind of data for the national parks to understand climate impacts and human disturbance. And what we were interested in is looking at the microbial and nutrient responses to these changes all within space and with time. Now, the one thing you have to understand for arid systems is that arid systems have always been understood to be water regulated and nitrogen limited. So it's water that controls when activity occurs. But the ability of the system to respond to the water is predicated on having enough nitrogen in order to do it. And so all of our work was focused on water and nitrogen and microbial response. And I'll show you a couple interesting patterns. So what we did along this elevational gradient is that we would go out twice a year, and we started building sites in 93. We finally got the money eventually in 95 and started collecting data in 96. But we would go out in January, being the coldest part of the year, and we go out in August, being the warmest and wettest part of the year, and we would collect these data from transects along this elevational gradient. And basically what this slide shows is those patterns of nitrate nitrogen, because all systems use nitrate. Now, one of the interesting things that you can see is that as we started out in 1995-96, for some sites, the amount of nitrate in the soil was pretty consistent among the elevational gradient. But over time, the system becomes chaotic. And you see these wide swings in available nitrate, irrespective of what's going on the previous year. So in some cases, and the red line was SG stands for Sotol Grassland. GS is Glen Springs, it's low elevation. CR is creosote, SG Sotol Grassland. Oak Forest and Lost Mine is the top of the watershed. So that's the gradient from bottom to top. And you can see that depending upon where you're at a particular year, you can get significant accumulations of nitrate in these soils. The question is, what's driving that? Well, you can also go out and look at the amount of microbial biomass carbon that's from the microbes in these systems, and we follow that over time. And what you basically see are these patterns across multiple years of what the biomass is doing, obviously in response to climate, but in response to what the nitrogen is doing also. And except you get these periodic pulses of enormous amounts of microbial biomass, such as happened in January of 2000 through January of 2002. That increase is due to caterpillars munching on oak leaves and all the poop going into the system, creating all this influx of nitrogen. And the system never went back after that. But you can see on the grassland, you see these periodic ups and downs and increases. And so the question was becoming, what's driving that pattern? We know that precipitation's changing. So when you look at Big Bend, what you see on that top graph is that in actual fact, there's not that much difference for the annual amount, at least at this time, the annual amount of precipitation that they were acquiring at Big Bend. It was, you had the highs and lows, but that wasn't changing very much. But what we discovered was changing was this figure. This figure, what was happening is that the interpulse periods between rainfall events were becoming longer and longer in the winter. And so something had happened to the winter storm tracks in which the interpulse period was getting longer. And if you notice how tight the interpulse period is early and what happens to it as we get in time, it's becoming more and more chaotic. It's becoming more and more chaotic. So obviously, precipitation and when precipitation was occurring has a major impact. And so as we started collecting more and more data off of multiple years, what we discovered was that in this RDA plot, moisture and ammonium is controlled by winter rains, and this is in the grasslands. The summer is where you get changes in nitrate, but because it's opposite, the vector is the opposite direction, it's what happens when it gets drier. So the summer is influenced by drought, the winter is influenced by rain events. And so we said, well, what happens if you start to change it? And what happens as these precipitation patterns change? And so what we did for 7 years is we watered the grasslands to see what would happen. Now, the interesting thing, and there's unfortunately none of my students who survived this are at Tech anymore. But you had to haul water uphill for a quarter of a mile. Okay, and we had to haul sometimes 500 to 600 gallons of water up to do this a quarter mile. Eventually, we got the park to drive up their fire truck and pump it up to some tanks that we put in there. But it took a lot of effort, and I lost a lot of graduate students that way, unfortunately. But most of them stayed. So I'm just going to give you one snapshot. Once we started to water, and we watered based on increasing precipitation 25% above what the previous month or two had received. So it was based on what was being received, and then we'd add to it. And we said, okay, we had a winter water event, a summer water event, and then a winter water event. So we had those and controls. So this is sort of a summation of some of that data, looking at a wet, dry, and very dry year. And these are all the microbial results. This is microbial biomass and functionality and enzymes. And these are 95% confidence intervals around these plots. So basically what you see on that left pattern is that the winter event doesn't have much of an effect. You don't see much of a change in a wet year, a dry year, or a very dry year. But it's the summer watering, the change in summer amounts, or the summer and winter amounts that were driving the system in terms of microbial response. Now, under a very dry year, it doesn't matter how much additional precipitation may occur eventually. It has no effect on that system. So there is a threshold below which you have no impact on a desert system. You simply is insufficient water to elicit any microbial response. But even in a very wet year, applying a little bit more water does have a benefit. So that's the watering event. At the same time, we were collecting soil temperatures. And what this shows is Glen Springs is our low elevation site. This is our Sotol grassland. And the upper line is the maximum soil temperature at 15 centimeters depth, not at the surface, 15 centimeters. And the point being here is that in these arid systems, at some point, the soil temperature crosses the air temperature max and stays there because soil in its bulk stores heat and maintains that. So all ecological systems store and maintain heat. And there's probably a thermodynamic profile for every ecosystem, whether it's urban or natural, as to how it stores and releases heat, which is characteristic of that system. And so the question we began to ask is that important? At the same time we were doing this at Big Bend, we happened to get on we were involved in a 5-year project at Fort Benning, Georgia, to look at ecological indicators for forest health for the Army. And we would collect similar kinds of data. Now, what we started to come up and discover is that when you looked at the difference in air and soil max temperature for a site which had a pretty good canopy and pretty good ground cover, you'll notice that the variability in the soil temperature was pretty low compared to what the variability was like for the air temperature. However, as you increased human impact, this is what happened to the variability in the soil temperature. It increased, and it increased substantially. And so we had, at this point, 5 years of Big Bend data along the transect. And so we said, okay, can we go back and look at the variability in soil temperatures in our transects to see how temperature was controlling microbial activity? And what we discovered was no microbial parameter was related to minimum temperature. No parameter was related to maximum temperature. However, microbial biomass carbon was significantly related to the difference. In this case, it was the monthly max and min soil temperature. And as that difference increased, the biomass decreased, irrespective of moisture. So we started then saying, okay, desert systems are water regulated, but then what role does temperature actually play? If you go to Google Maps and you look at, this is that little triangle Y, or that little Y on that road up here, is rice tanks on the Glen Springs Road at Big Bend. If you look at this system and sort of move your Google map to that, you'll see this in the desert. And someone said, what the hell are those? What those are our experiment. That's what you see. What we've done is, since 2006, we have passively modified the daily temperature variability by shading the soil surface to reduce solar input. And so we have two sets of projects that are going on. This is Natasha's Van Gestel's. We started with this project back in 2006, so we had to cut them so the creosote bush showed through. This sits about 15 centimeters above the soil surface. And Michael Hyman, one of my master's students, we set this up about a year or so ago. We have continuous monitoring of soil carbon efflux from these systems, and we're shading them the same way. So that's what you see. So we're looking at how this change, how we can modify DTR, the daily temperature range, and understand what the microbes are doing in response to that shift in DTR. So how is it shifting? Well, Natasha, as you know, graduated, is now in northern Arizona. She left Sunday, but here she's measuring some creosote bush for photosynthetic activity. But what we've done with our plots is that we have increased the night temperature a bit, and we've decreased the day temperature max, and so we've decreased DTR. And the question is, what's the response? Well, this is sort of a summation of Natasha's 4 years of data collection to a single graph. That's the problem with a lot of dissertation work. You spend a lot of time for one graph. But basically what it shows is that if you look at the microbial biomass carbon and microbial biomass nitrogen in spring and summer, there is higher biomass nitrogen and there's higher biomass carbon when you decrease DTR. And this is in the same situation where there's no change in soil moisture. It's simply a decrease in the stress those microbes see in response to temperature. So one of my other master's students, Nirmala, started looking at what really the microbes are doing. Now, the problem with Big Bend, and it was fortuitous, is we went into a major drought. So the same plots have been going since 2006, and now we have a situation where, as you get into 2010, 2011, things are becoming drier and drier and drier and drier. But what happened is the system flipped in terms of DTR. Initially, there was not much difference in DTR at 15 centimeters. That's this part of the graph. Most of the DTR effect was at the surface, is what we see. So some of it may have been translated down, but not a lot. But we could still see a reduction in DTR. As we went into the drought, notice what happens to air DTR in that response. And then look what happens to DTR in the soil with no moisture. But once we started going into the drought and started coming out of the drought, the system flipped. And now, same sensors, same plots, everything's been the same. Now what we have is as a consequence of dry soils through this whole period, we're seeing the DTR lower down, where there's almost no difference in DTR at the soil surface because it's too dry. You cannot have an effect. So drought plays a major role in how soils store and lose heat. So this year, Diana, one of my master's students, we started a project in agriculture. We said, okay, if DTR really has that effect, you should be able to use it in agriculture. So what we did is we put erosion control blankets out in a cotton field. And we planted cotton and then put the blankets back on. We have them all censored up to know what soil moisture and soil temperature doing. And she's working on the data, but one of the things that I think is important is what happened to the cotton under those blankets. And what you found was you got better emergence and growth of cotton when you reduced DTR. You didn't change anything else. All you did was change DTR. We got a better stand. In fact, we got the best stand establishment at the USDA, at the Tech farm, with changing DTR compared to what they would experience in a normal cotton field. Now the question for us is, if that's the case, no farmer is going to put this across most of West Texas. So how do you change DTR effectively? And what it's going to be based on is bed design. How can you change bed design to modify that? And then the other part of the issue is, if you change bed design, can you shift when farmers plant cotton? And so that's part of what we're looking at. So to bring us to conclusion, we know for Big Bend that DTR has been changing at about, 0.18 degrees centigrade per decade. It's been decreasing. Globally, it's at 0.12 degrees centigrade per decade, globally. So the arid systems are decreasing DTR faster. And I think what you have to realize then, for all of us, whether it's a human system or a natural system, while in arid systems, while moisture may regulate activity, the magnitude of that response either is enhanced or reduced by DTR. It does matter what those soils are doing, but changing climate variability through DTR is going to have an unknown effect on how systems respond to climate variability. One thing I haven't told you, because we're still trying to ferret out exactly how this is occurring, are soils that had reduced DTR actually respire more carbon off, even in an arid system. So there's a negative feedback to those systems. And that's the unintended consequence of climate change then for an arid system. Thank you.